I'm a plant physiologist. Particularly I look at how plants interact with the environment and how plants react to stressors in the environment, particularly pollutants such as radioactive pollutants. One of the main things that ultimately drives almost everything that all of you are doing are potential effects of radioactivity on the biosphere. Okay? Everybody here is in some way part of a radioecological kind of paradigm. In the summer school we've been looking a bit more, my contribution has been to try and introduce the students to some of the effects of radiation that people predict happen in the field. Some of you I know from hydrofoam are working at sort of how things might diffuse through solid rocks if they leak out of a waste repository. One of the reasons why we're worried about we have to calculate what might come out of a waste repository in 8,000 years' time, whatever it is, in case some of those radionuclides might at some point interact with the biosphere. Okay?
So we're in the area near Chernobyl, Neil, so what is it we need to look for?
What we're looking for is just a plant species that's got a reasonably fleshy leaf, because we just want to squash out the kind of internal contents of the plant cells, which will be mostly pigments, but there are lots of things in the Chernobyl Exclusion Zone which is slightly difficult to do that with, so all the pine trees that you can see, their needles are very difficult to squash. There's quite a lot of shrubby plants that have quite leaves that aren't very fleshy.
So ferns, for example, what are they like?
Ferns are not so good because most ferns have this kind of divided up leaf, so it's a bit difficult to squash them. What we want is a reasonable sized leaf that's slightly fleshy that we can squash things out of. So something like this is a bit of a small example. We might find a bigger example with bigger leaves, but that's fat hen, but that's got quite nice fleshy leaves that would be quite good to try and squeeze all the internal contents out of. The grasses also, it's very difficult to do. The grasses tend to be quite fibrous, so it's difficult to squash things out of them. So what we need is another example of fat hen that's got a bit of a bigger leaf on it. So
so this one here, for example? Yes.
This one will be good. So you can see if we just take the water off that, you can get that's quite a nice soft fleshy leaf that's not too fibrous.
Have a feel.
So,
Oh yeah.
Yeah. And this is quite common in the Chernobyl Exclusion Zone, and so if we remove some of those leaves we ought to be able to squash those leaves nicely and express all the contents from the inside of the leaf and see if we can extract the pigments from that.
Excellent, let's give it a go.
Okay.
Others of you are working on various aspects of transfer and so on, but all of those things are potentially important because of the effects that ionising radiation might have. So the exercise that we did yesterday was just to start to introduce to all of you some thoughts about how radioactivity can affect the biosphere and how it can have adverse effects on organisms of one sort or another. Okay?
So basically what we want to do is to get a strip of the plant pigments across the bottom of here, so that we can then use this as a little kind of chromatographic system to separate out the different molecules that we've extracted from the leaf.
Okay. And what will actually cause those molecules to separate? Why would they separate out?
Because of the different molecular weights that they have. They're basically the molecules are different sizes. So the smaller ones move faster through the gel, and the bigger ones move more slowly.
Some of you are working absolutely with effects on different types of organisms. Others of you are working on various aspects of transfer and so on, but all of those things are potentially important because of the effects that ionising radiation might have on the biosphere.
Lay the leaf along there, and then if you get a coin that's got a smooth edge, as it happens, one of Her Britannic Majesty's 2p coins has a nice smooth edge. If you do it 2 or 3 times, you just get a greater volume of pigment on the strip, or alternatively you just get a bigger mess. But actually that's probably not too bad. I've just split the silica gel in the middle, so we might get kind of 2 bands moving out.
But one of the things that effects comes back to is the radiolysis of water. Okay, so ionising radiation by definition can ionise molecules.
living things are mostly water. You are about 82 or 83% water. One of the main things that the ionising radiation interacts with in you is water. So radiolysis of water, the splitting of water due to ionising radiation is a really important thing for starting to understand the effects. Okay?
So what's happened is we've extracted all the cell contents, and in these bottles then we've got a small amount of ethanol. So most of what we've extracted there will be pigment, so you can see it's green. So most of it's chlorophyll.
Yep.
But there are other things in there as well. And if we stick this in here and let the ethanol run up the strip, we should be able to separate everything else out from the chlorophyll and see what we've got in there. So you literally just strip slightly wide, so
So just trim it down.
I'll have to just trim it down just so that it can actually fit in the bottle. There will be things that we've squashed out of the plant cell that won't dissolve in an organic solvent, and they'll just stay where they are. So we'll get a bit of a kind of a mucky grey strip remaining. But the things that are soluble in organic solvents, which most of the pigments are, so most of the pigments are embedded in membranes, so they'll dissolve, not dissolve, or they'll be picked up by an organic solvent like ethanol. Oh, sorry.
Yeah.
It's a reasonably thick one, so we'll see what happens. Yep, that's okay. Those yellow and orange pigments are carotenoid pigments of one sort or another, and you get quite a lot of carotenoids in carrots, hence the name. But those carotenoid pigments are kind of used to help protect the systems that carry out photosynthesis in the plant. So some carotenoid pigments help to protect the photosystems by absorbing light. If there's too much light coming into the photosynthetic systems, some carotenoid pigments will absorb extra light. But one of the other things that carotenoid pigments do is they mop up some of the dangerous products that can be produced during the process of photosynthesis. So photosynthesis starts with the splitting of water, and the splitting of water releases protons and electrons and also oxygen. Now hopefully, if everything's going well, what happens is those electrons are fed into an electron transport chain. But if things aren't quite optimal for the plant, sometimes those electrons might not feed into the electron transport chain. They might get a chance to recombine with the oxygen that's produced by the splitting of water.
And what would the effect of that be then?
The effect of that is to produce superoxide, which, as the name suggests, is highly oxidising. And that superoxide then might start to oxidise the proteins that make up photosynthetic machinery and all those sorts of things. So if it can avoid it, the plant doesn't want to have superoxides flying around.
Why is it a problem if those things get oxidised then?
Mostly, if they get oxidised, it damages them, it changes their structure, it'll then change how they function. Essentially, they kind of get burnt, if you like, by being in the presence of lots and lots of superoxide. But so one of the protection systems that a plant has to stop all those
superoxides flying around, if they are produced, is some of these carotenoid pigments. So some of the carotenoid pigments can help to mop up superoxides that are accidentally caused during the process of photosynthesis.
That's fine. So what we did was we squashed some leaves. Okay, so we got some chromatographic strips, literally just some kind of thin chromatography layer strips, and we squashed some leaves onto it. Anybody remember which plant leaves we squashed onto the strips?
The lilac?
Lilac. Okay. What we did was we looked for a plant that occurred reasonably regularly and hopefully in low dose and high dose areas, and we squashed the pigments onto a piece of thin layer chromatography paper, and we did that just literally by rolling a coin over the leaf to kind of squash the leaf onto the chromatographic paper. And if you use a smooth edged coin and roll it over the leaf, you can kind of squash all the pigments out of the leaf. Okay.
And those yellowy and orangey pigments, they have a number of different uses in plants, but they're very commonly used as antioxidants. And some of them find their way up the food chain into animals, and animals use them as antioxidants. And both plants and animals have a variety of antioxidants that they use, but carotenoids are used in both plants and sometimes in animal systems as one component of their kind of antioxidant system.
Ethanol. What's the significance of the ethanol? Why not water or something else? Why ethanol?
It's an organic solvent.
It's an organic solvent. Why is that important?
It dissolves the cell membrane.
It does actually, it's true. We didn't mention that yesterday, but actually ethanol does kind of destroy the cell membranes. The cell membranes are made out of lipids, so the ethanol does destroy cell membranes. And what's in the membranes in the plants?
Pigments are released.
Pigments. That's right. The pigments are released because the organic solvent, the ethanol, breaks apart the membrane. It releases the pigments. The pigments are lipid soluble. The pigments are not floating around free in the cell somewhere. They're in membranes. Okay, and they are solvated by organic solvents like ethanol. So when we put our little strip, dip it in ethanol, the ethanol moves through the strip and it drags with it the different pigments. Okay. And it drags the pigments with it because the pigments are lipid-soluble molecules that will dissolve in an organic solvent.
Yeah.
So you've taken these samples, you've run them in this way.
Yeah.
What did you find? What was the result that you found?
Well, we basically in all the samples we looked at particularly where we went at Birkhoff, there's quite a lot of lilac growing, which I think the lilac probably comes from people's gardens from when it was a actual active village some time ago. But across the Western Trace we found lilac plants quite regularly in dose rates starting at about 0.5 microsievert background all the way up to 20, and we ran lots of these strips, and in all of them you get lots of yellow carotenoid pigments coming out, and it just doesn't look like there's any effect on the carotenoid pigments. When you line them up, you wouldn't know which ones had come from a high dose rate area and a low dose rate area. They've all got nice yellow and green bands, and they all look pretty similar. Yeah. So that's beginning to separate quite nicely, and it is possible to you can look at these just by eye, but it is actually possible to use absorbent materials and take the pigments off here and put them in a spectrophotometer and really quantify exactly how many carotenoid pigments you've got. But mostly you can see by eye that even in the areas of high background radiation there's plenty of carotenoid pigments there, and it doesn't look like the high dose rate is using up the carotenoid pigments because it's producing lots of reactive oxygen species by splitting water up.
Yeah. A great demonstration that field science can use fairly simple techniques to still allow you to study something very important.
Yes,
the effects of radiation in the environment.
And it's difficult to get exactly the same amount of pigment on every strip, and actually they run a bit faster and slower with different temperatures and all that kind of stuff, but you can see visually pretty quickly that if you go across the Western Trace from a lower to a higher dose rate area, all the plants seem to have plenty of carotenoid pigments in them. So my money would be on the fact that where people have measured changes in antioxidant concentrations, changes in the concentration of carotenoid pigments in things living in the zone, it's for reasons other than the radiation, probably.
So birds need not be concerned about the colour of their plumage from now on.
No.
Right.
If there are indeed changes in bird plumage, or if birds that need carotenoid pigments to make particular colours of plumage, if birds are having difficulty with those sorts of things, I suspect it's not because of the radiation, it's because of the predators or their lack of food or abundance of food or other ecological factors rather than the radiation itself. What we saw is that actually in quite different dose rates we went across the areas of the Western Trace, from low dose rate areas to some higher dose rate areas, and you can see that lots of the pigments in plants that might be an indicator of whether they're stressed or not, they're just exactly the same in the higher dose rate areas and the lower dose rate areas. And so I think as far as the plants are concerned, we didn't see, at least in the little bits of work that we did, any immediate evidence that the plants are being particularly stressed by the radioactivity that's in their environment.
So can then anybody remember what the pigments were that we thought were separated there?
Green is chlorophyll.
The green is chlorophyll. Plants are absolutely stuffed full of chlorophyll. Okay? And chlorophyll is what they use for photosynthesizers and so on. But what was the significance of mentioning photosynthesis as far as this exercise was concerned? What does photosynthesis do? What kind of reaction is photosynthesis?
Also radiolysis water.
Photosynthesis starts by splitting water. The splitting of water in photosynthesis is called photolysis. So actually photosynthesis does what radiolysis does, but it uses energy from light. Okay? And the chlorophyll helps the plant split water in a process of photolysis, which is exactly analogous to the process of radiolysis that occurs when you have ionising radiation coming through a cell.
Some of those pigments are used by plants to protect themselves from some of the things that can be produced by environmental stresses, such as radiation. So we've measured the concentration of pigments in different dose rate areas of the Chernobyl Exclusion Zone to see whether we can find any evidence that the radiation is causing the plants any stress. And we hope that that helps the students understand some of the kind of physiological things that people talk about when they talk about the effects on organisms.